Hello everyone, my name is Samuel and this is my term presentation and the title is Designing Synthetic Antimicrobial Peptides as Antimicrobial Agent. Here is the outline for my presentation, which includes introduction, experimental suggestion, result and discussion, and the last conclusion. As for introduction, AMP or antimicrobial peptide as a solution to bacterial resistance. The global public health threat of antimicrobial resistance has led the scientific community to highly engage into research on alternative strategy to the traditional small molecule therapeutics. And the first documented failure of antimicrobial therapy in humans due to acquired microbial resistance was reported in the 1940s for penicillin, which occurred only a few years after its commercialization. Two factors are identified as the reason. The first is the generalized use and misuse of antimicrobial therapies and the insufficient discovery of new antimicrobial therapies. And therefore, antimicrobial peptides have been identified in various species, ranging from bacteria, frogs to mammals, including humans. They are actually the first line of host defense against pathogenic infections and are a key component in the innate immune system. AMP consists of up to 50 amino acid residues, which depending on their composition, size, and conformation, can be arranged in different groups, such as cationic peptides, non-cationic, aromatics, etc. And they have broad spectrum activity against many gram-negative and gram-positive strains, as well as fungi. And they can bind to the anionic bacterial surface and can integrate into the cytoplasmic membrane, making pores to kill the microorganism. And the strategies behind the design of AMP for antibacterial application is due to its ability to interact with bacterial membranes or cell walls. And the requirements essential for peptides that targets the membrane of organism is, first of all, a positive charge of peptides, which is critical to enable them to interact with the negative membrane. Furthermore, the hydrophobic portion of the peptides is essential to enable the insertion of the membrane into the membrane's hydrophobic core. And also the helicity of the peptide and the non-polar phase of the alpha helix structure are also vital for successful insertion into the membrane. And for natural AMPs, commonly have 12 to 50 amino acids and are highly cationic with net charge between 2 to 9 and also amphipathic. And starting with this general design, recent strategies for AMPs include the using of machine learning, modeling tools, and combination libraries. And it is called bioinformatic tools. And a classic application of those tools is in the use of artificial neural network, qualitative matrix, and support vector machine, which will be used in this work. And computer-assisted AMP design is useful for estimating the desired biological activity from the primary peptide structure. And most of the naturally occurring antimicrobial peptides have rather long sequences, which can compromise their application as drugs due to the high cost of production at industrial scale. Therefore, my goal here is to identify a short active fragments as a base to develop the AMPs, uh, and is promising due to its relative ease of synthesis and low cost production. And the idea is to propose a sequence of amino acids by employing some criteria from natural antimicrobial proteins or peptides, such as positive net charge, hydrophobicity, and helicity. After coming up with the design, it is necessary to characterize the synthetic AMPs and understand the mechanism. And in this work, I would like to try a template AMPs, which is not an enzyme, but an AMP itself, which is a cationic peptide called C18G, which is a synthetic alpha helical peptide derived from human platelet factor 4. And this peptide is found to be antibacterial and is active against Salmonella. And the peptide sequence is as follows. After choosing the protein sequence as my template, the C18G sequence will be analyzed using 4 open source online servers. DPABBs to design the peptides with antibiofilm activity, CellPPD to design the cell-penetrating peptides, HeliQuest to predict the wheel projection and hydrophobic moment, and the last is ToxinPred to evaluate the toxic potential. And here is the result. So in this work, I selected the antimicrobial peptide C18G, which has a broad spectrum and with positive 8 cationic charge from 7 lysine residues in the sequence. And here I choose to design the peptide with only 10 amino acid residues as an effort to find a similar or more effective antimicrobial peptide, but with shortened sequence, which will be easier to synthesize with higher purity. And through the analysis, some peptides from C18G show high potential as antimicrobials. The cell PDD generate 9 peptide sequences, and all of them here are predicted to be cell-penetrating peptides. Meanwhile, the DPABB server also generates similar 9 peptide sequence with 10 amino acid residues, and out of 9, only 6 of them were predicted to be biofilm active, and 3, the rest are predicted to be inactive. Therefore, these 6 peptide sequences were both active against biofilms and also have the cell penetration abilities. And these peptides will be the ones that are worth characterizing and evaluating further regarding their antimicrobial properties. And we can see that they are cationic with the charge ranging from positive 4 to positive 5, which is an essential feature for antimicrobial activity. And one thing to be noticed is that normally a very high positive charge, for example more than positive 9, is actually can backfire and result in no antimicrobial activity and even toxic to mammalian cells. Meanwhile, our peptides only have positive 4 to 5. And this is important because one of the concerns for synthesized peptides is their toxicity. We expect them to be able to kill bacteria, but also at the same time do not harm the cells. And ToxinPred was used to calculate the toxin potential of these peptides And the result shows that none of the peptides are toxic. They are all non-toxic. And the helical wheel projection was analyzed by using HeliQuest, and it reveals more features about the peptides. Usually, a wheel projection is expected to show a helix formed by the hydrophobic and apolar amino acid concentrated on either one side of the helix. And this was the case for our peptides, as can be seen in the picture. The hydrophobic residues is presented in yellow and apolar in silver, while positively charged residues are in blue and the polar residues are in violet. The diagram revealed that the hydrophobic region, yellow, and the positively charged region, blue, in all six peptides are gathered at the same place for, for the same group, but each group are separated from each other on different sides. These two group separations are important for antimicrobial activity because they can have two types of interaction with the cell membrane, which is electrostatic, in which the attraction force drives peptide membrane interaction, and hydrophobic or apolar, involved in the peptide insertion in the membrane. And this wheel diagram leads to the conclusion that all of them have well-defined segregation of polar and lipophilic phase, which means their alpha helix seems to be amphiphilic, which can be the basis of their reduced antimicrobial action. Therefore, all the six peptide sequence met all the requirements essential for them to target the membrane of microorganisms as predicted by our bioinformatic tools, such as having a positive net charge around 4 to 5, having a low hydrophobic ratio, and they are also short amino acids, only 10, and also having a alpha helix secondary structure, and also predicted to have the cell penetrating and antibacterial activities. Therefore, they are good candidates for and have potential for synthesis and further antimicrobial assays. Thank you very much for your attention and see you when the pandemic is over.